Grade 12 biologists, prepare to have your mind blown. This week we're looking at the nervous system, and trust me, it's not what you think. So first thing that I want to point out is that what I'm trying to do with my introductory slide here is to equate brain function with computer code. And they're not exactly the same, but the similarity is that computers operate by using what's called binary, ones and zeros. This is switches that either allow electricity to go through, that's a 1, or they don't, that's a 0. And in a very similar way, the cells in your brain make those same sorts of circuits like switches by either allowing a signal to go through them or not. Ones and zeros. So the first thing you need to understand is what those specialized cells of the nervous system look like. And believe it or not, this contraption here is a giant, elongated, wacky-shaped human cell, and it's called a neuron. This is what the cells of the nervous system look like. And essentially, they look kind of like wires, but what's really cool about them is that integrated into those wires is also those decision-making switches. We can receive signals through these branches called the dendrites, and then we can send signals down what's called the axon. That's the true wire part. And then at the end of the axon, it can branch out into many axon terminals. And in reality, these axon terminals would be interfaced with the dendrites of the neighboring cell. And so this cell body is where you'll find the nucleus, which can perform all those regular everyday functions of any other neuron. For many of the axons, they're covered in another type of cell. So these little guys that each have their own nucleus are called Schwann cells. And together, these Schwann cells that are covering the axon are called the myelin sheath. And as an analogy, that's essentially like the insulation on a wire. When we're talking about a brain, what we're actually looking at is a network of interconnected neurons. And so here you can see dendrites interfacing with axons of two neighboring neurons. Now in your brain, there's billions of these neurons all interacting. And so we can receive signals here, send them down these wires, the axons, and then they reach this physical gap from one cell to the next, and we call that gap a synapse. And this is where the decision making happens. Either that signal will be passed on to the next neuron, or it won't, a 1 or a 0. Now, another important part to realize is that these cells are so wonky shaped and elongated that oftentimes they're associated with these glial cells, which are kind of neighboring cells that help to nourish and keep these other wonky shaped neurons alive. And so the synapse is really where the interesting part of these neurons tends to happen. This is the decision making. And so before we get into how exactly that synapse works, we need to cover what this concept is here of an action potential. I know that historically, when you've heard about electrical activity in the brain, you've probably thought of it a lot like an electronic device. In fact, if you get an electric shock, your muscles will contract, and so you might think that your nerves are kind of like electrical wires, but they're not. Okay, this is not a copper wire with electrons flowing through it. It's actually quite different. They transmit signals from one place to another using what's called an action potential, which essentially is an electrical change caused by the flow of ions, electrically charged ions. When we're talking about potential, we're talking about a difference in electrical potential energy from one side of the membrane to the other, based on differing concentrations of charged ions, not the flow of electrons. And in fact, it's much, much slower than the flow of electrons, but still pretty wicked fast. And so let's have a look at what that kind of looks like. In a nutshell, when we stimulate a neuron, what we're actually doing is opening up ion channels. And so when an axon is at rest, it's slightly negatively charged on the inside relative to the outside, which is slightly positively charged. In other words, there's more positive ions out here and less positive ions and more negatively charged ions in here. But when we stimulate it, we open up these ion channels, allow positive ions to rush in, and we essentially flip the polarity of this membrane. And so we call this depolarization. And when it is relatively positively charged inside the axon versus outside the axon, we say that we've reached action potential, which will be a difference of exactly plus 40 millivolts. And what that's going to do is it's going to stimulate the neighboring ion channels to start to open over here. So when an action potential begins, it starts to get transmitted, it starts to move. And immediately behind that action potential, we're resetting the trap by pumping out or by releasing potassium ions into this space and then reestablishing that resting potential again. So in this way, the signal can move from one part to the next, and then leave behind a reset neuron to receive a new signal behind it. So this really is the movement of chemicals, ions, that's going to be sending signals. And when those signals reach the end of an axon, so here you can see the axon terminal, there's something entirely different waiting for them there. These vesicles are waiting, and inside of those vesicles is a special chemical called a neurotransmitter. Neurotransmitters basically are like hormones, but rather than being released into the blood, they're instead going to be released into the synapse. And so here you can see multiple action potentials reaching this axon terminal, which is stimulating multiple vesicles to start releasing neurotransmitter into this synapse space. And when it does, and I know it's a little bit small in this diagram, but I hope you can see this neurotransmitter acts as a ligand to a receptor on the postsynaptic neuron. So this is the dendrite of the neuron next door. And of course, what that's going to do is either open an ion channel on it, allowing ions to flow in and creating an action potential in there, or those ligands may be closing those channels, which would be preventing it, right? We've got ones and we've got zeros, and in fact you can have hundreds of these interconnections giving different conflicting signals to the neighboring neuron, and if you get just the right mix of the influx of ions into the neighboring neuron, it can fire. And so this is going to be an all or none response. Either it's going to get stimulated enough or it's not going to get stimulated enough to continue that signal on. And in this way, it works kind of like a computer switch. Yes, no, 1, 0, all, or none. And so here are a few examples of some of these neurotransmitters, and you may have heard of some of these words before, like dopamine and serotonin and endorphins and GABA. Acetylcholine is another fairly common one that we talk about. You may be surprised to see things like adrenaline and noradrenaline on the list. What I want you to think about is that we don't only release these neurotransmitters into the synapse space. You can also release a neurotransmitter at a nerve ending that directly interfaces with, let's say, a muscle. In some cases, you can even release stuff into the bloodstream, like what we learned last week the hypothalamus is doing through the pituitary gland and the posterior pituitary gland. So there's a little bit of complexity here into how these neurotransmitters work. But for the most part, know that these are the chemical signals that we use in the synapse. And we're starting to learn to manipulate and learn to use these for medications to even help people deal with mental health issues. And so throughout your body, you've got this system of wires. And what those wires really are the axons of neurons. And if you trace them all back, these hugely elongated axons make their way back to one of two places, either just to the spinal cord or all the way back to the brain. And so we call this the central nervous system, and we call these blue ones the peripheral nervous system. And yes, that's right, a single cell can have axons that can extend from your brain all the way down to your toes. But let's be really clear about the difference between a nerve and a neuron. Each one of these nerves is actually a bundle of very, very many axons from very many neurons. So if we look inside of a nerve, we'll have some sort of outer coating for strength. Inside will have some fatty tissue as insulation. It will have blood vessels running throughout it to keep these cells alive, and then you'll have these bundles of multiple insulated, myelinated axons carrying signals down to a particular location from the spinal cord and or from the brain. And so the peripheral nervous system consists of two main types of neurons. Those that are bringing information towards the central nervous system, and those that are bringing the response signals away from the nervous system towards effectors. Sensors, effectors, and the integrators all work together to have stimulus, integration, and response. And so any neuron that's bringing information towards the central nervous system, we're going to call those afferent neurons. And afferent neurons are generally also called sensory neurons. Okay? It's bringing information towards. The decision making, okay, the understanding of that signal, is carried out by interneurons, which are found exclusively in the central nervous system. And believe it or not, both your brain and your spinal cord can make those sorts of interpretations and decisions. Generally, the responses generated from our spinal cords we call reflexes, and we call the stuff happening in our brains thinking. Okay, we're understanding, though not all of that is conscious. Finally, we can send information from the interneurons out to our effectors via what we call efferent neurons, moving away from the central nervous system, and we'll call those sometimes motor neurons because they can affect things like our muscles and create movement. So quickly, let's look at how this works in our simplest pathways, a reflex arc, where we don't even need to necessarily use our brain for the response. We have some sort of sensory input. Maybe this is touching something really hot or really sharp and pointy, and that's going to send a particular sequence of action potentials down an afferent neuron. And when it reaches the spinal cord, which is what you see here, inside of your spinal cord you'll have some interneurons, and here's a single interneuron with its axons leading to two different locations. If this signal is of the right quality to warrant an immediate response, let's say that's super hot or super sharp, okay, lots of pain, it will take only one or two interneurons to say, get away. And so it will send that signal directly to an efferent neuron, which is going to make its way straight back to your muscles and tell you to pull away as quickly as possible. At the same time, it will also send that signal to your brain, but that's going to take you a little bit of time to process and understand what happened. By the time you realize what's gone on, your hand has already moved away. We call that a reflex. So here it is depicted again in full color. You've got your afferent neuron carrying the signal to your spinal cord. Here's that interneuron that is immediately detecting emergency and time to respond, and it can directly tell the efferent neuron to pull your hand back. It will also send that information to your brain, where now it's going to have to go through many hundreds or thousands or even millions of neurons in your brain until you can actually process what just happened. And that'll take a little bit of time, maybe even more than a full second. And so here's the general lay of the land. The central nervous system is connected to the peripheral nervous system, and it's receiving both inputs from afferent neurons and giving outputs through efferent neurons. The peripheral nervous system can be then subdivided into the sensory or afferent division and the motor or efferent division. And generally when we're talking about response, some of those we're thinking about and have control over. We call that our somatic nervous system, the one that we can control consciously. And here's the autonomic one, which is running all the stuff that we're not thinking about, all of those automatic responses. And those generally come in two flavors. They can either be sympathetic or parasympathetic. And all that means is if it's parasympathetic, it's your general regular housekeeping, maintaining your heartbeat, keeping your regular breathing. But in the sympathetic, this is when there's danger, and we kick into a whole other set of autonomic responses that we sort of feel as our flight or fight response. And of course, all of this is perpetually giving feedback back to the central nervous system. And so let's get into a little bit more detail about exactly how those action potentials travel through a neuron. And so here, this diagram is showing you a little bit more clearly how these ion channels work. The blue ion channels represent sodium ion channels, and the orange ones represent potassium ion channels. And you can see that when this axon is at rest, it's sitting at -70 millivolts. What that means is that if you put an electrode both inside and outside of that axon, you would detect that it's slightly more negatively charged inside this cell than it is outside. And the reading on your voltmeter would be -70 millivolts inside of the axon. You can also see that there's a high concentration of potassium inside the cell and a high concentration of sodium outside of the cell. So we're going to learn how we set that up and get it ready to fire, but that's going to be the perfect conditions so that when we open up these sodium ion channels, sodium can come rushing in. And as you can see, wherever that sodium is rushing in, we're changing the polarity of that membrane from -70 all the way up to +40. So we call -70 millivolts resting potential, and we call +70 millivolts the action potential. And as you can see, once we reach action potential, the neighboring sodium ion gate opens as well. So we call these voltage-gated channels, meaning that what causes them to open and close is particular electrical charges. Let's have a closer look. So here's how we might create a graph depicting what an action potential looks like. Here we can see the axon is at resting potential. And here we can see some stimulus. Now, if that stimulus allows some ions in, okay, a slight depolarization of the membrane, we can maybe graph that, but it's not always going to result in an action potential. The voltage-gated sodium channels are only going to open the neighboring ones when they reach -55 millivolts. We call that the threshold potential. That's the number that when it's reached, the sodium channel next door, boom, pops open. And when that happens, we reach rapid depolarization. In other words, the sodium ions are rushing in. And once we reach this action potential of +40 millivolts, they're going to respond again. This time they're going to slam shut. And now the potassium ion channels are going to open at action potential. And so those potassium ions will come rushing out. And then we can use sodium-potassium pumps and some ATP in order to reestablish those initial concentration gradients. We'll have a closer look at this in a moment. So let's have a look at it again. Here you can see an axon, and we're looking at the inside of it in cross-section. And so I'm going to represent my ion channels then with these symbols here. This is an open potassium channel, and this is a closed sodium channel. Both of these are voltage gated. At resting potential, the sodium ion channel you can see is closed, and at resting potential, the potassium channel is open. And so by resting potential, I mean an actual number, and that is the potential difference between the inside of the cell versus the outside. So if we connected this voltmeter, it's going to read -70 millivolts, meaning slightly more negative inside than outside. Another difference that we would notice is if we looked at the chemicals that were present. And in that regard, we would certainly notice that there would be a high concentration of sodium outside of this axon and a high concentration of potassium inside. So we've got not only an electrical gradient, but we also have a chemical gradient. And if you remember from learning about the mitochondria, we call that an electrochemical gradient. And it's a very, very powerful one. So this electrochemical gradient is really encouraging these sodium ions to want to come into the axon. And that's because it wants to go from high to low concentration, as well as positive being attracted to this negative region. Likewise, these potassium ions would like to get out of here in that they've got a concentration gradient. However, that electrical gradient is encouraging them to stay. And so in reality, even though that potassium ion channel is open and those potassium ions are free to go in and out as they please, overall they tend to stay inside of the axon because that electrical gradient is keeping them there. So where does the electrical gradient come from? Well, the axon is manufacturing these negatively charged proteins, which are maintaining that negative 70 millivolts. And so now you can see this setup maybe in a slightly more realistic way, where these sodium ions really want to come in but can't. And these potassium ions are free to go but don't, because there's this electrical gradient keeping them there. During a stimulus, which is generally going to be some sort of an electrical change in the axon, these voltage-gated channels are going to reverse their configuration. So those sodium ion channels are going to open and those potassium ion channels are going to close. And you can imagine that in that moment, sodium is going to come rushing in along its electrochemical gradient. And since those potassium ions can't leave, we very quickly increase the charge inside of this cell all the way up to positive 40 millivolts. And when we reach that value, we call it the action potential. That action potential is going to be the signal to reverse the configuration of these ion channels once again. So we're going to close the sodium ion channels in that moment and open the potassium ion channels, and look at what the situation we've got now. It's positively charged inside relative to what's now negatively charged outside. So we have an electrical gradient to get these positive ions out, and we have a high concentration of potassium ions and a low concentration of potassium ions outside. So just as quickly as we depolarized the membrane, we can repolarize the membrane temporarily with potassium ions. But if you recall, we're still not at resting potential yet. We need to get those concentration gradients back. We don't want potassium outside of the cell and sodium inside. We want it the other way around. We want these sodium ions out and those potassium ions back in. So when we repolarize the membrane, sometimes we even overshoot that negative 70 millivolts inside as the potassium ions rush out, but we still aren't where we want to be. How do we get back to this configuration? Well, the answer is we're going to use sodium-potassium pumps to reverse this concentration gradient. And so a sodium-potassium pump uses ATP, and this is why it takes so much energy just to think. In fact, about a quarter of your calories every day go into the electrical processing that your brain is doing. This organ is incredibly hungry for energy just to reset the resting potential in the axons of your neurons. And so here's what it looks like. It's going to take in 3 ions of sodium, bind to ATP and get phosphorylated. And in that moment when we release that energy from the phosphorylation, boom, we're going to change the configuration of this sodium-potassium pump and release the sodium outside of the axon. Now that it's open to the outside, potassium ions are free to bind, and that is going to cause a conformational change that's going to knock off that phosphate and allow the potassium to enter. And so in reality, what we're going to get is what Drake is demonstrating to you, this energy-using pump that's getting sodium out and bringing potassium back in to reestablish resting potential. And this is probably the slowest step in an action potential, is to get the concentration gradients right. But when I say slow, we're still talking milliseconds to get this thing back to where we want it to be. Thanks, Drake. And so here's my demonstration of it, where you can see that those sodium-potassium pumps work until we have the right concentrations. And once these concentrations are established, we stop using energy, we stop pumping. And so there it is. Resting potential at negative 70 millivolts. If we reach negative 55 millivolts from some sort of stimulus, all of those sodium ion channels open, sodium ions rush in. And then we want to repolarize by closing those sodium ion channels at plus 40 millivolts and opening up the potassium ion channels. Outrush the potassium ions, and immediately those sodium-potassium pumps get to work and return us back to resting potential with the correct ion concentrations. Take a minute to pause the video and see if you can explain what's going on in this picture here that's showing you the state of these voltage-gated channels at resting potential with a small amount of stimulation, at threshold potential, at action potential. Here's our sodium-potassium pumps at work, and then back to resting potential. And so there it is. The action potential is simply the depolarization of this membrane, which causes the neighboring voltage-gated sodium channels to open and passes the action potential along. Now we can actually make that go significantly faster with this kind of insulation. So here you can see an axon, but on the outside of it, wrapped around it, are these Schwann cells, and together these are making that myelin sheath. The key is that there's these nodes or gaps between the Schwann cells where we can alter the concentration of these ions. And so essentially when an action potential flies in here, it's going to then make its way across that Schwann cell and depolarize at the next node. And in this way, we kind of skip the area in between. This allows the action potentials to be transmitted hundreds of times faster in a myelinated versus an unmyelinated axon. And so there it is. Action potentials going down axons. When they reach the synapse, they're going to release neurotransmitter, and that neurotransmitter is then going to stimulate the neighboring cell. And so the key here is, if we reach threshold potential in the neighboring cell, negative 55 millivolts, then it will fire. If we don't, no firing. And so ultimately, the circuitry of your brain is just a complex network of neurons, which we call the neural network. And the way that they interface and the strength of those interactions is going to determine how they process information. And so this is one of the main goals of brain science today, to try to uncover what those circuits look like. And we don't really have great tools yet to look at an active human brain to figure out how these things are working in this closed black box. But we're starting to. And one of the most important projects of our time is called the Human Connectome Project, which is trying to look at exactly just what the raw wiring looks like. And let's admit it, even if we figured this out in its entirety, and here's a preliminary map of what we're starting to come up with, just knowing the wiring of something this complex doesn't really tell us how it works. There's a lot of work to be done. Here you can see some actual slices of imagery through a real human brain. And using this MRI technology, we can get a pretty clear picture of what's in there. And you can see there's different colored regions. You've got this white stuff in the middle, and you've got pink stuff around the outside edge. And the white stuff in the middle is generally where the axons are, and the pink stuff is where the cell bodies are with all of their dendrites doing all of the processing and decision making. So we've got wires and we've got processing areas. One of the ways we're starting to get insight into how these things work is looking at simpler creatures than us. Here is a much clearer picture of nearly the entirety of what looks like the neural network of a fruit fly brain. Pretty incredible stuff. But overall, what we've managed to do is at least start to divide up the brain into different lobes that do different sorts of jobs. There you can see an outline of some of the main tasks that we feel are associated with different parts of the brain, and we know this by kind of poking and prodding it and electrical stimulation of it and recognizing what happens when different parts of the brain get damaged. Here's a really important area called the sensory motor cortex, where the frontal lobe and the parietal lobe meet along this groove. And where they meet, we can actually detect that different sections of the left and the right side of the brain control and receive information from different parts of the body. And so here's an artist's rendition of the sensitivity of each of these parts based on how active these neurons are, how much innervation there is. And so you can see we've got a lot more sensitivity in our hands and in our feet and in our mouths than we do in the rest of our limbs. Another interesting thing to look at is what's inside of that brain. And while most of that processing and thinking and most likely our consciousness is located is probably in this cerebral cortex, this big green region here, but there's all these other structures inside of the brain doing all sorts of different functions too. And when you look at the brains of other living creatures, like for example this rat, they've all got those other parts, less so that cerebral cortex. It seems like this is one of those things that kind of separates the higher thinking type organisms. We believe that this is the more primitive part of the brain, and that's evidenced through our evolutionary ancestry as well. You can see that a fish is almost entirely composed of what we call the mid and hindbrain, whereas that forebrain, very little. Amphibians have a slightly more developed forebrain, reptiles an even more developed forebrain, and finally we get into birds, which have it fairly large and well developed. Ultimately, it's the mammals that have really mastered this part of the brain, and it's so large that it's actually covering up the mid and much of the hindbrain as well. And in a human, it's almost the whole thing. So if you look at the evolutionary tree of brain development, there's a massive spectrum of different mammal brains. We're certainly not the only ones with a highly developed and highly folded cerebral cortex. In fact, you might be surprised to see the brain of an elephant, which is just by mass significantly larger than ours, is also more highly folded and has more surface area, implying that it can do very complex tasks. And have a look at the brain over here of something like a dolphin. And of course, these brains are separated in evolutionary time by millions upon millions of years. Who knows how they see and process the world, but you can bet it's in fairly complex ways. Here you can see a size comparison of brains as well. And there's still that question out there of, does size actually matter? And is bigger really better? Perhaps signals move slower if they need to travel over greater distances. There's all kinds of factors that we're still trying to weigh out and figure out. But you can see that a rat brain in its entirety is only about 1 centimeter long, whereas ours is significantly bigger, but certainly not the biggest. And so biology is working very hard right now to use all sorts of experiments to understand the inner workings of that brain. One of the coolest and most controversial fields is this field of optogenetics, which I had you look at during the genetics unit. But it's not the only tool that we have or have had in the past to understand how the brain works and functions. There's all of the social sciences as well, such as anthropology, sociology, and psychology, which are also trying to use the scientific method to uncover how the brain works, mostly by looking at our culture and our behaviors. And so that's all I've got for you today. I hope that whets your appetite to learn more, because there certainly is a lot more to learn when we're talking about neurons and the nervous system. But I hope that helps. Thanks so much.